Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 15th video of chapter 2 of Immunology, and this is part 2 on the Neutrophil Extracellular Trap. For short, we call them as the NETs. In the first part of this video, I've told you that NETs are web-like structure that is composed of chromatin, histones, and antimicrobial protein. And the NETs, they are released by the activated neutrophils, and the NETs are responsible for trapping and killing the pathogens. I've told you that the formation and release of the NET is called a NETosis, and during the NETosis, the neutrophil undergo a unique form of cell death where it releases its DNA into the extracellular space, forming a meshwork of fibers that traps and kill the microbes. I've told you that for the activation of the neutrophils, there are a variety of the signals. For example, the interaction of the pathogens with the neutrophils, that can be one of the ways by which the neutrophil they can get activated. The inflammatory cytokines, that can be another way where you can go for the activation of the neutrophils. So the first step in the process of the NETosis, that is the activation of the neutrophils. The second step, that is the production of the reactive oxygen species. And in the last video, I've told you that in the neutrophils there is a specialized enzyme which is known as the NADPH oxidase, which is responsible for the generation of the reactive oxygen species like the superoxide anion, the hydroxyl radicals. And these reactive oxygen species, they are actually responsible for an oxidative stress within the neutrophil, and that is very crucial for initiating the downstream signaling events. So this is actually this oxidative stress which is initiating the downstream signaling events that we discussed in the last video. Then I've told you that the third step in the NETosis, that is the nuclear decondensation. The fourth step, and in the nuclear decondensation, I've told you about the an important enzyme which was the peptidyl arginine deaminases, and this particular PADS was responsible for the citrullination. And during the process of the citrullination, the arginine residues in the histone proteins they get converted into the citrulline residues. And when they convert into the citrulline residues, that is going to alter the structure and charge of the histones, thereby promoting their dissociation from the DNA and facilitating extrusion of the DNA into the extracellular space. Then I've told you about the mixing of the nuclear and the granular components. That was the fourth step in the process of the NETosis. And I've told you that after the decondensation of the chromatin mixes with the granular components within the cytoplasm, including the histones and the antimicrobial proteins. And this mixing process that actually prepares the components necessary for the NET formation. Now, in this particular video, I want to focus on the fifth step of the NETosis, which is actually the rupture of the nuclear and the granular membranes. Now, if you are interested in mixing the chromatin with the antimicrobial proteins, the chromatin is present in the nucleus surrounded by a nuclear membrane. The antimicrobial proteins, they are present in the granules surrounded by a membrane. So if these membranes, they are there, you will not be able to go for the mixing of the chromatin with the antimicrobial proteins. For that to achieve, for mixing to achieve, you have to rupture the nuclear and the granular membranes. So let us talk about this. The nuclear and the granular membranes of the neutrophil rupture, allowing the mixed content to spill into the cytoplasm. Because if you are interested in the mixing of the chromatin, the histone and the antimicrobial protein, you have to rupture both of the membranes, the nuclear one and the granular one. First, I'm going to focus on the rupture of the nuclear membrane, how that is achieved. Now, there are a variety of the theories or a variety of the phenomena that has been reported which are responsible for the rupture of the nuclear membrane. The first one, that is the reactive oxygen species induced disruption. What happened is that the reactive oxygen species, as I told you, they are produced during the activation or after the activation of the neutrophils. So the reactive oxygen species, they are directly or indirectly contributing to the breakdown of the nuclear envelope by inducing the oxidative damage to the nuclear membrane components like the lipids and the proteins. and this is one of the most established phenomena that the reactive oxygen species, they are responsible for the rupturing of the membranes. This is one of the mechanisms by which you can rupture the nuclear membrane. The second one is the calpain-mediated cleavage of the nuclear pore complexes. Now these calpains, they are calcium-dependent proteases. That means that for their activity they need this calcium. So these calpains, they have been proposed to cleave the components of the nuclear pore complexes. If you look at the normal functions of these nuclear pore complexes, they are responsible for regulating the transport of molecules between the nucleus and the cytoplasm. For example, the proteins in and out of the nucleus, or the other materials in and out of the nucleus. Now, the disruption of these NPCs, that could lead to the breakdown of the nuclear envelope and the release of the chromatin into the cytoplasm. So these NPCs, they are very important for maintaining the structure of the nuclear envelope. If the calpains, they are going for the disruption of the NPCs, that could lead to the breakdown of the nuclear envelope and release of the chromatin into the cytoplasm. And I would say this would be the decondensed chromatin, because the arginines that have been replaced by the citrulline now. The third mechanism that has been proposed, that is the chromatin swelling and pressure. Now, as the chromatin decondenses within the nucleus during the NETosis, it undergoes significant swelling due to the loss of the histone DNA interactions. Now, keep in mind one thing: the DNA that has been condensed to an extent that it can easily fit into the nucleus of the cell, and the nucleus is occupying a very small portion of the cell. Once the histones, they are gone, what that means is that the chromatin that will be in the loose form, that is going to exert pressure on the nuclear membrane, and when that pressure is exerted, that actually leads to the you can say breakdown of the nuclear envelope. So this is another mechanism that has been proposed. Now this, as I've told you, this is going to exert mechanical pressure on the nuclear membrane, eventually leading to its rupture. For example, if the chromatin is 2 centimeter, for example, the histone is going to condense this into 1 millimeter, for example. So 1 millimeter can easily fit into the nucleus. It is not going to exert any mechanical pressure on the nuclear membrane. But when the histones, they are gone, it again moves from 1 millimeter to 2 centimeter. That is going to exert pressure on the nuclear envelope, and that will eventually lead to the rupture of the nuclear envelope. The fourth mechanism that has been proposed, that is the actin polymerization and the nuclear deformation. Now, it has been suggested that the polymerization of the actin filaments that may generate mechanical forces that deform and rupture the nuclear envelope. So you can expect any of these mechanisms that would be responsible for the breakdown of the nuclear envelope and the release of the decondensed chromatin into the cytoplasm of the cell. Now that is one thing. Now if you release the chromatin from the nucleus, the decondensed chromatin from the nucleus, but if the granular membrane is still intact, you will not be able to mix the chromatin with the components of the granules, which are the antimicrobial proteins. So the next thing you have to do is you also have to rupture the granular membranes. Now how that is achieved? Now, as I've told you, one of the most established mechanism, that is the damage to the membranes by the reactive oxygen species. So just like the nuclear envelope rupture, the reactive oxygen species induce oxidative stress within the neutrophils that could lead to oxidative damage to the lipid bilayers of the granular membranes, compromising their integrity and eventually causing the rupture. So as the reactive oxygen species, they are damaging the nuclear envelope. By the same mechanism, they can damage the membranes of the granules, and when the membrane of the granule, they are damaged, they are going to spill their components into the cytoplasm. That is one mechanism. Another mechanism is the enzymatic cleavage of the granular membrane proteins. Now, if you look at the neutrophil granules, they contain a variety of the proteases. For example, the elastases, the proteinase 3, the cathepsins. I have detailed videos on these enzymes, and these proteases, they may target proteins within the granular membrane. causing their cleavage, destabilization, and the release of the granular content into the cytoplasm. Now you can compare this with the NPCs. Once we degrade the NPCs, the nuclear envelope, it gets destabilized and it ruptures. Same is the case with these membrane proteins, or these granular membrane proteins. So once these proteases like the elastase or the cathepsins, they cleave these membrane, when they cleave these granular membrane proteins, they lead to the destability of the membrane, thereby leading to their rupture and the release of the granular content into the cytoplasm of the cell. Again, the actin dynamics and the cytoskeletal remodeling, as I've told you, actin filaments just as they exert mechanical pressure on the nuclear envelope, they can also exert pressure on the granular membrane, contributing to their rupture. Now, the sixth step in the process of the NETosis, that is the extrusion of the nets. We have ruptured the nuclear envelope, we have ruptured the granular envelope, the granular membrane. The components, now they are mixed, the chromatin, the histone, the antimicrobial peptide, now they are in contact with each other. You have mixed them by the rupture of the granular and the nuclear envelopes. Now this time for the extrusion of the nets. Now the ruptured neutrophil releases its DNA along with the associated histones and antimicrobial protein. As I've told you, now they have been mixed. Now the mixture is actually released into the extracellular space. Now this extrusion process forms the nets, which is actually the meshwork of fibers, as I've shown you in the last video, and I'll show you that in the next slide as well. And this particular net is responsible for trapping and immobilizing the pathogens. Now, once the extrusion of the nets that have been achieved, now it's time for the maturation and stabilization of the nets. You have to make a stable net. Now, once released, the extracellular DNA fibers, they undergo further modifications. For example, there is methylation, there is acetylation of the DNA, again, there is citrullination of the histones, and all of these processes, they promote the stability and the functionality of the nets. Now, these modifications, they also enhance the antimicrobial properties of the nets, because if you are having a stronger structure, you can trap the microorganism or the pathogen very well. You can make a stronger interaction between the antimicrobial components and the pathogens, thereby effectively killing that particular pathogen. So stability, that is the key. And the last step, once you have made a stable net, you can actually go for the microbial killing and the immune modulation, because the meshwork of the net that have trapped, they have killed the pathogen by physically restricting their movement and by exposing them to the antimicrobial proteins and enzymes associated with the chromatin. Now, why this net is important? Why are we discussing the net in so much detail? Because while the net played a crucial role in the host defense, the dysregulated net that has been implicated in a variety of the inflammatory and the autoimmune diseases like the rheumatoid arthritis and the thrombosis. So that means we need a controlled release of the nets, a controlled function of the nets, because if there is dysregulation of the net, that can lead to a variety of the diseases. Now let me summarize the whole story. This is the neutrophil, this is the bacteria, you can see over here. These are the granules present in the neutrophils. This is the DNA of the neutrophil. These are the histones. These are the myeloperoxidase, elastase. It would be just some of the example of the antimicrobial components over here. So if you can see over here, summarize the whole story. You are going to activate the neutrophils when the bacteria, for example, that interact with the neutrophils. You are going for the production of the reactive oxygen species. You are going for the nuclear decondensation. Then you are mixing the component of the nuclear and the granular components. You are going for the rupture of the nuclear and the granular membranes. You are going for the extrusion of the nets. You are going for the maturation and stabilization of the nets. Then you are going to kill the microorganism and the immune modulation. You can see over here the bacteria that has been trapped by these chromatin fibers, and when that is trapped, it can actually come in contact with these antimicrobial components like the myeloperoxidases or these elastases, thereby killing this particular bacteria or any other pathogen. So this is all about the nets, about the NETosis. So if you like the video, please subscribe to my channel, hit the notification bell. If you have any question, you can ask me in the comment section, and do share it with your friends. I'll see you in the next video.